And right now, Stephanie Hughes is up at the University of Alaska Fairbanks doing her PhD in his lab. So there's a strong connection between Los Angeles and University of Alaska Fairbanks, if you didn't know that they had that connection. So Todd's background, he's got a bachelor's and master's degree at Villanova. His PhD was in pharmacology, toxicology in Virginia, in the medical college in Virginia, and then he got his DBM at Wisconsin. And then after getting his PhD, DBM moved up to Barrow, Alaska. If you know where Barrow is, it's in the world practically. It is the northernmost spot in North America. And 7 years? How long were you in Barrow? 10 years in Barrow. If you've ever been to Barrow, that's a long time to be in Barrow. He liked it. I was in Barrow for 3 weeks, and that was long enough. And then after working in Barrow, and actually his first experience with marine mammals was at that point. We were doing a lot of stuff with the Native natives that lived there, Alaskan Eskimos, and their interaction with marine mammal populations. So his first experience with marine mammals started with Barrow. Then he moved to the University of Alaska Fairbanks, where he's been for a long time and has done a lot of work with toxicology, marine animals, and how they interact with humans, because humans are also in Alaska consuming marine animals. So you got to have a connection between the marine animals itself, the environment, and the human population, and that's partly what he's been talking about. So happy to have Todd here for a seminar to hear, and he's got obviously some interactions with the lab, which we'll probably talk about. Oh yeah, I'm gonna brag about.
I'm going to try and mix this up a little bit. It's not going to be a highly technical seminar because I'm going to get a little philosophical, and I'm going to make sure the connections that Jim just introduced you to are apparent. And that might be this is a little bit of a recruiting mission too. So take it whatever best suits you. We are very much interested in this One Health perspective in Alaska, and now we're developing this with Colorado State University. So when I put CSU up here, I apologize, that stands for Colorado. Because we're starting a Department of Veterinary Medicine that's going to be articulated with Colorado State University, so we're going to start training Alaska residents in veterinary medicine in Fairbanks. And we're going to base it on this One Health concept. So I want to introduce you to it. For that reason, and plus some of your students are coming up and working with us on this as well. So we think it's pretty exciting. This is a art that was done by a Chupik artist on Nunivak Island. And I'm going to come back to this once I walk you through some of our concepts here. Jim's right, most people don't know where Barrow is, and when I was recruited for this position, my friend said, don't look at a map, just listen to me. So Dan Mulcahy was down here in Anchorage giving me that advice. And of course my wife immediately went over to the bookshelf and started looking for a map of the West. But 10 wonderful years up there, and introduced to some amazing cultures, amazing animals. And we hesitantly moved this far south to the Fairbanks, and we don't think we can go much further south. We're definitely an Arctic family. This is called the subarctic, so it's okay. We're often referred to as the interior, but I'll have you know the School of Fisheries and Ocean Science is based in Fairbanks. So in Alaska, everyone thinks they're coastal. And if you don't believe that, look at these rivers. They're all salmon rich. And so salmon really connect us with the ocean along with other organisms. And so it's funny because there's a marine culture actually in Fairbanks. It's really one of the organisms that's harvested for food in Barrow. And so this is the bones that remain from some of the harvest. The gray whale stranded here, and so I'm just trying to show you the food web goes from Cabo to the Arctic. Okay, so this is a gray whale that's allowing multiple bear families to come in, partake in. They normally don't do this. They're rather aggressive to one another, but when there's this much resource available, there's an amazing amount of tolerance. And we put a helicopter in the air, and because of these stranded whales in this bone pile out here, there were 60 bears in the area. That becomes a human health issue, kind of like directly. So we tried to make sure they stayed out here at Point Barrow and didn't come into town. But here's your California connection, and the bears thank you for the food. I want you to recognize that pinnipeds are food in Alaska for people, for various wildlife, and just want to point out that she goes out and breaks little airs and provides seals for her young, and just it's graphic for a reason, just to show you that they are incredible hunters. I can put more footage and so on up here of how they hunt walrus, how they hunt blue whales. These animals, these polar bears can kill very large organisms. Watching them kill and pull a blue whale out of the hole is awe-inspiring. They are amazing predators, and if you don't know, they're very closely related to the grizzly bear, the brown bear. They can actually breed and produce grizzlies. Yeah, grizzlies. But this is what Coca-Cola wants you to see, right? And this may be what you would want to see, but they are beautiful animals. They're very charismatic. They're very interesting, but they are predators, and they enjoy marine mammal diet. So what I'd like to do is talk to you a little bit about these paradigms that we think in. I'd like to review a little bit of the history I've had up there because I really want to give credit to the people who have helped, and it's a privilege. Being in this profession is a privilege, and I'm thankful all the time, and my wife is too. We get to raise our kids in this environment. It's really a great privilege. I'd like to talk to you about some of the tools we're developing for our research that have impacted students from lost landing. And then a little bit about sentinel species, because I can't escape without doing that. So when we talk about environment, disease agent, and the host, we need to talk about the epidemiologic triad. That's the way we want to get people to think. You've probably seen this before. But John Harley, if you know him, he was at Santa Cruz, worked in the Costa lab for a while as an undergrad. He helped me put this together along with Carson Hoofer. We're trying to put a marine spin on this where the ocean, microorganisms, the disease agents, and hosts such as killer whale, we want to better appreciate this interaction. We want to think about the interactions of living and non-living components. We're trying to develop this better in our work. And again, I point out that here's something else that enjoys the occasional pinniped. I'm going to keep emphasizing that because we see seals, walrus, sea lions as the middle of the food web. Often I hear people down here talk about them at the top of the food web. That may be true here, unless you're a great white shark, killer whale. But seals are really important. They're kind of like a big fish. So you got to think about that, them as a food item in Alaska. So how we tie this into the One Health concept is it's the American Veterinary Medical Association that said we're going to have an integrated national strategy to better train veterinarians and to get more involved with public health through this paradigm. That is, we're going to recognize ecosystem health, human health, and animal health overlap. The vet schools were criticized by the National Academy of Science for training too many small animal clinicians and not enough big thinkers. And we're going to follow their advice, and we're going to produce Alaska residents who have this perspective. Now, they can go do small practice, but they're going to be the eyes and ears of surveillance for when there's disease issues that need to be addressed. I could give examples where large animal veterinarians and other veterinarians have been the first one to observe a foreign animal disease or a disease of concern. And we need to have a better public health response in that respect. Department of Defense, Homeland Security, all all very concerned about the lag phase from the point of detection until we actually do something. So think about West Nile virus, influenza, monkeypox, all these things you probably heard about in the press. So it's One Health is not just a feel-good, well, how can we all kumbaya together, right? They actually think it's a way of improving public health and being more responsive. So I come back to our Chubik artist, and I want you to recognize the smiling Chubik Eskimo surrounded by terrestrial and marine wildlife. All I did was gave them our proposal, which was we're going to study nutrients and contaminants in your foods and how food processing alters them and what it means to your health. That's all we gave him. He came up with this. And I thought it was interesting that he took a Venn diagram approach too, how things overlap and interact, and he put the Chubik at the center. And I thought that was really interesting. And so in discussions with him, he goes, "Oh, I forgot the plants." And I said, "That's okay. They take care of the plants." So I thought it was very interesting, and I'm really proud of this artwork. It became an icon of our research. We wanted something that people could identify with other than classic scientific ways of representing what we were studying. And this was very popular in the community. So, and here's our way of looking at it. So, when we talk about One Health context in Alaska, it's real important to recognize we link wildlife and human health in three major ways. One is we've discussed the subsistence diet. That's hand and mouth, that's pretty obvious. Here is a bowhead whale that's been harvested that's going to be cut up for food, and there's price per hour. One of our colleagues in Barrow, and that's my wife Carla. So this will get processed for food. And then here's obviously salmon. And then over here is probably bearded seal that's drying. So subsistence diets are obvious. Wildlife as a model for humans may not be as obvious, but at the University of Alaska Fairbanks, we're rather famous with NSF and NIH for hibernation models, and DOD, Department of Defense. And the reason that is occurring is because we have animal models that hibernate at -2 Celsius. Ground squirrels freeze. They go to -2 Celsius when they're hibernating. This absolutely fascinates health professionals and people in defense because obviously it helps in dealing with trauma, organ transplants, heart attack, and stroke. How do these tissues have the resilience to deal with going -2 Celsius in a mammal? And every 30 days they warm up when they're hibernating. And one theory is they warm up to dream. In other words, the brain has to say, "Oh yeah, I'm still alive." It's fascinating. And so these animal models are very important in physiology research and potential biomedical processes for improving human health. Wildlife is an indicator of human risk. I'll talk about that when I present the sentinel species idea to you. So think of how wildlife can tell us what's going on out there in the ecosystem that might be advantageous. So this is how we see the world in our lab at the University of Alaska Fairbanks. What's important to recognize is this is also important in outreach, education, and community involvement. And so here I am in Sitka, Alaska, at Mount Edgecombe High School, which has Alaska natives from all over the state attending this high school. And here we're using stranded cetacean as a teaching opportunity where we talk about ocean and human health, the marine ecosystem, and then anatomy and physiology of this animal. This has become really popular. We call it necropsy demonstrations, but what we do is we go into the mission to really talk about the marine environment and how important it is to humans, and teach them about the adaptations these organisms have undergone to be effective in the marine environment. And over here is a harbor seal. And in this case, this was high school students. In this case, this principal canceled all classes, kindergarten to 12th grade. The whole student body came out on the basketball court, and the hunters and I butchered these animals and necropsied these animals at the same time. I call it a butchery. That he's taking food and telling them about how they harvest the animal and what they take for food. At the same time, I was talking about scientific sampling and how we do health assessments. It was very powerful. It really meant a lot to the community. And so we're asked to do this more. And up here is one of my favorite images. This is a couple miles 60-ton whales out of. And they paddle out, and it's a hand-thrown harpoon. This is their bowhead line. They'll be behind this is the bowhead surface. It's a stealth hunt in this case. So I wanted to just point out how we interact with these communities. It's all about fish in our state. Pathways of nutrients and contaminants, commercial, subsistence, recreation. Fish are really important to us in Alaska, so I just want to emphasize that. So we do our research, we do our outreach, we do our education, we do our community involvement all at the same time, and we're really proud of that. So this is, what Jim said, the end of the world. My boss used to say, no, Barrow's not the end of the world, but you can see it from there. Barrow is the highest point in the United States as far as north, and it's right at the Chukchi Beaufort Sea. It's a remarkable place. It's an area that is highly productive. The Inupiaq are there for a reason, because incredible amount of resources are there. So this is just the upper quarter of Alaska. This is a county known as the North Slope Borough. I'd like to point out it's the size of Minnesota. Okay, so, and it had, when I was there, it had about 8,000 people total. So we were the species that was probably the lowest population. There was 500,000 caribou in there. So it's a remarkable place. So I was a research biologist there, but they wanted a DPM PhD, and as I walk through this, I think you'll see why. So they said, we need someone who will study bowhead whale health assessments. That's still going on today. They still do that. And they still hire veterinarians to work with biologists to do that. And that was important for the International Whaling Commission. They also decided they needed a veterinarian to address environmental contaminants. Immediately in 1995, I was doing on health. I didn't know that's what it's called. But they wanted to address animal health and human food security at the same time. So I was responsible for determining if these environmental contaminants were having adverse effects on the animals, but at the same time I had to discuss how they were as food items for people. It was fascinating, and we've never changed that perspective. We also had to deal with food security and safety in response to mortality events. Caribou, moose, seals, something that would be 100, 1,000, thousands of dead animals, and we would have to respond to figure out what caused the mortality event, but we also had to determine whether it was a food safety issue. And we ran into things like Brucella. I'm sure people know about that here, but that's a zoonotic agent that's not fun to get. And then radioisotopes. People are always releasing radionuclides, so as a toxicologist, I'm always in business because there's always a radioactive accident somewhere, and regardless of how far away it is, it gets people excited in the wrong way. And so, always dealing with radionuclides. It obviously involved remote, high north communities. Had to learn how to work with communities of 100 or 200 people who may be 90% reliant on local foods. That's a very different way of looking at how people eat than maybe down here. The other part of the job was humaneness and efficiency of hunting. They wanted to improve their hunting. Time to death they wanted reduced, and they wanted the number of injuries that did result in mortality reduced. And so we worked really hard with them to improve hunting methods. And a veterinarian is usually someone who's responsible for appropriate handling of animals, including how they're euthanized or killed. And so this was a big part of my job, and it's still a big part of the job up there. The basic biology, this was absolutely critical, and I think this is where people here who are being trained in biology should recognize this, is that my training, as Jim said, I didn't know anything about marine mammals when I went up there, but fortunately they had 3 people there who were well trained in marine biology or wildlife biology. Without that, I would have been lost. And so I want to point that out, that was critical for the veterinarian being there. What was important was I could interpret all of the data we had in the proper context, that we knew about the population status and trends. If you really think about it, how often do you have that advantage? Often we're working in mysterious situations where we really don't know the status of the population or the stock or the species. Because we had such an intensive bowhead whale program, very important in research and teaching, and so we've never left that. So for 20 years, we've been doing that. It's been very helpful. So why did we go to the University of Alaska Fairbanks? The National Institutes of Health decided they wanted to fund an infrastructure grant at UAF. So this is how we ended up there, environmental agents of disease. And so then we took these concepts and we've been applying them to our program since. Ecosystem health, ocean and human health, NOAA and NSF, and One Health. So get to the people in our group. We have a postdoc, we have a research professional. Some of you may know the Castellinis. Been working at Pinnipeds for 30 years, 40 years. Incredibly valuable person out in the lab. She has much more experience than I do, so it's great. And then here's some examples of the students, and I'll highlight these guys at the end because they come from down here. And just want you to know there's a California connection. Funding, again, is diversified because of the various perspectives we have. We have state funding and federal funding, and industry has actually started funding some of our work. So obviously, it involves a lot of teamwork, and I always joke, I don't know if I'm there or there. I think I switch back and forth, but the idea is that we have to fight the current as a team. Some of you may have romantic ideas of Alaska, and that's great. Our tourism folks love that. But we're no longer dealing with such logistic issues that we have to mush everywhere. This isn't our field station. We can do a little better than that. And as much as I'd like to be romanticized as the rugged individualist wildlife veterinarian out there tackling moose, not using any chemical mobilizing agents or anything, that's sort of not true either. This is actually the group that first climbed Denali. And Diana Campbell's related to them, and she provided me the news. So I'd like to talk about some of the tools we're developing and how some of the groups down here have helped us. It's this blood-soaked filter paper technique that I think is something that's really going to revolutionize field work for a lot of us. With EPA and the Department of Interior, with a human health group, said, can you help us develop rural monitoring programs for disease agents? Can we find a simplified and robust technique? And what it's called is the Rural Alaska Monitoring Program. And so I'm going to introduce you to a technique that you might want to adopt, or you might want to help us validate it. This is Pat Curry from University of Calgary, and these are the filter papers we'll be talking about, these filter paper strips. Here's an example of a hunter-killed animal and how you can collect the blood by slicing the jugular vein and applying these filter papers to that free-flowing blood. You can also do it live capture where you pull it out in a syringe and you can soak the filter paper. So this is a technique that has been used in, I don't know if you know, but like in infants with a blot of blood. But here we can get a little, here we can get a little more volume. So the idea is you cut it here after it's been blood soaked, and this strip will absorb about 100 microliters of blood. It's fairly standardized. And so this is really important in the standardization of the sampling technique. So what we do with that is we can air dry it. And then you can stick it in an envelope, and it's preserved. So in the field, you don't have to worry about carrying around whole blood. You have to keep it frozen. And that's why it's so valuable, because we can archive it that way, we can transport it that way, and that's why those agencies wanted us to help develop this technique. Some of you know Francis Golan, so we validated this with harbor seals from the Marine Mammal Center, as well as bottlenose dolphins from Sarasota Bay. So this is coming out this month, or is out now, maybe, in the Journal of Wildlife Diseases, where we were able to validate this for mercury. Now, working with Colorado State University, we're going to see if we can do additional chemical surveillance using this technique. So you may have a use for this if you think about some of your field operations or how you want to archive blood. It very much limits the logistical challenges and the cost of sampling blood. It also allows biologists and hunters to do it in a more convenient manner. So we were able to use something called the direct mercury analyzer for this. No processing was required. The funding agency said, yes, we want to move forward with this. It's this simple, it really is. So here's that filter paper. We cut the strip of blood off. This actually fits perfectly in the direct mercury analyzer, and you analyze the whole thing. Boom. So everyone's very pleased with how this has worked out. Here's the data for it. This is the whole blood, so we have matched whole blood as part of the validation study. And then down here is the filter paper with the whole blood on it. And as you can see, we've got a very nice relationship between the two. The dark are the dolphins and the clear are the harbor seals. So we've been asked to move along on this and develop it for other species from mercury, but also include other analytes. So this unbiased chemical surveillance is what we're going to try next. And what we're going to do is use a mass spec technique that Dr. Dooley at Colorado State is developing. And what we'll do is have various extraction methods that based on differences in solubility. We'll go from water soluble to lipid soluble extractions, and we'll put it on the liquid chromatography mass spec. That he has two of these down there. And we're going to start off with northern fur seals. We got funding to do this in northern fur seals, but then we're going to expand to other species. It's not just about environmental contaminants. We're also seeing it work for us with serology and PCR. Our colleagues in the same issue of neurologic diseases have published this for looking at various infectious agents, serology of the infectious agents, that is looking for antibodies to those agents. We're now looking for DNA of the pathogens in the blood. So you can see how this is going to become a valuable sampling technique. The key is blood soaked filter paper is allowing for multiple uses, and this is going to be real important. So again, serologic and PCR based. John Harley, who I'll get to later, he's going to try and do this with messenger RNA. I think he's dreaming, but that's what graduate students are for, right? Take on these challenges. Don't feel limited. So we're going to see how far we can go with our PCR-based methods. So again, one sampling event. We can look at multiple environmental agents from one sampling event. This is being funded by the departments here at EPA because they're really concerned about climate change, and they want a One Health perspective on this. So we'll be analyzing some of the strips in real time, and some of the other strips are going to go into an archive. So that's what they would like to develop. If you've ever had to store blood over many, many years, frozen, it's a real difficult thing because of the cost and also because when freezer failures occur, we were shocked. We got contacted by some Scandinavians and said, can we please use this technique on humans? And that's where we slammed on the brakes and said, wait a minute, because obviously handling human blood is very different because of safety issues. But also because we weren't sure if we should be interpreting data from humans yet. But they want us to develop it for that. So where we're going with this is we're going to develop these techniques for how we can improve our antibody detection, the PCR-based molecular diagnostics. Again, the DNA-80 worked great for mercury. But what surprised us was they immediately asked us to do additional elements, essential elements and other non-essential elements like cadmium, and other toxicants. The LUA concentrating methods are important because some assays you can't have dilute plasma. And I'll show you what I mean here in a minute. So the LUA might be too dilute. Well, we found a centrifugal concentrating method that seems to work, at least for macromolecules. So this is how it will work. This is the filter paper. Remember, with the DNA-80, we just put that directly on. But if you want to look for antibodies or anything like that, you cut it up and then you put it in this phosphate buffered saline, and then you can use the centrifugal concentration to get down to maybe the original concentration in the plasma. And we did some preliminary data on bottlenose dolphin blood for toxoplasma gondii. And here is the serum titer. So this is serum that we got directly from the animal. And then we did that procedure I just showed you where we concentrated the remained negative. So that was a very important finding. And here I've highlighted where, in red, where between the serum and the eluate technique, we were within a twofold dilution. If you know anything about titers, they're twofold. So 2,048, the next one is 4,096 because you're having the sample. That's how the titer works. So this actually worked pretty well based on the antibodies to toxoplasma. I assume everybody here knows about toxoplasmosis here because otters. Well, this is a serious disease up in the Arctic as well. It's actually passed on through the food web. It's not cat litter and cats. It's actually food web based. We also decided to do preliminary work on organochlorines, and a vet student from CSU got an award to come up and work with us in the summer, Lily, and she did this in terciops, again, in bottlenose dolphins, and her preliminary data was encouraging that we can use the filter paper technique for organochlorines. However, we need to find lower detection levels, and now we're working with a group in Environment Canada, Gary Demir, who's a very famous environmental chemist, at least in the Arctic. He's going to help us see if we can improve this with lower detection levels. But the data was promising for things like PCBs. So we've been asked to go on into essential elements and other non-essential elements. These are not as direct as doing the total mercury, but we'll see. And so it looks like my sabbatical will be to do an unbiased surveillance of these organic chemicals at Colorado. I think this is encouraging for field work and hope people can see how archiving these and using these can be beneficial. So enough marketing of cellulose paper. We're pretty excited about it because human health people and wildlife management folks and environmental management agencies are saying they all want this. So it's an example of One Health. And even though it's a simplified technique, I hope you appreciated that it took sophisticated techniques to analyze the filter paper. Does that make sense? So don't focus on, filter paper, it's so easy. No, it's the technology that we can apply to the filter paper that's exciting. And you can see I'm kind of animated about it because I am sort of super geek when it comes to this kind of work in the lab. And our lab people are just thrilled with this because they know what it's like to go out and cut blood and have the blood tubes and keep frozen. If this will work, it'll really help a lot of field work. So when it comes to sentinel species, some of you may have already heard this Stella Sealine story, but I really would like to emphasize it because it's one of the major issues we're facing. When it comes to One Health or ecosystem health, sentinel species are really an important part of that. This is now combined with the filter paper approach. NOAA has decided that we're going to try the filter paper approach on sterile sea lions. And if you want to talk about remote sampling, I'll show you here in a minute where they have to go. It's important to recognize sentinels are usually recognized as something that integrates the system. For the sterile sea lion, it's fish consumption for us. It's the pathways of contaminants. So they represent an integration of fish, you might say, in that area. But it also applies to commercial and subsistence fish. So there's, again, that human health connection. Sterile sea lions are a management concern. They're also very cute. So put a cute picture in. They're cute pictures coming. It's not all about data. Not sure what she's mad about, but I think probably the photographer got to close. But Lori Rhea is the lead on this, along with our colleagues here from NOAA and Fish and Game and at the university. We're focusing on the females and looking at how fetal mercury concentrations are distributed over the Aleutian Islands. Mercury is important because of in utero exposure. So here's the Aleutian Islands, and well, they change the designation all the time, Jim. Now they're called a distinct population segment. We used to call them stocks, that was wrong. But they get called all kinds of things. Now they're called DPS. But there's a magic line here, and it's the eastern DPS and the western, so you can see this group's doing well, and this group's not doing so well, indicated by population status. Here. So I want to remind you, Kenneth, I'll remind you, mercury bioaccumulation, biomagnetified. That's why he's in the front row. humans and other fishing animals, and it crosses the sun. And we also found out it's really high in fog. So again, congratulations on your NESO board. That's great. Well, it's not great that mercury's high in fog, but getting the award is good. There's a lot to be learned about mercury. You think it's well studied, but we really don't understand it very well. So this Western group, through these publications, has consistently had high levels. And it didn't really spark interest until the Castellini paper came out. And again, remember, it's a distinct population segment. And so Dr. Rhea and I and some others decided we needed to look at these guys. And we don't sample them as they're coming out. I don't mean to show you this image. But what I'm trying to emphasize here is they are whiskered, they are covered in hair. That means this was developed in utero. I think that's pretty good evidence. That it was developed in utero. And so this means this was what we can look at the mercury exposure in these guys when they were during gestation, right? When they were in utero. Very important because that's when most of the adverse effects might take place. Luckily, they hold on to this for 2-3 months. And so Noah and others go out and they can sample these animals, these young skeletal sea lions, so we can get at their lanugo and determine what their in utero exposure was. I don't know how many of you have ever had the opportunity to work on skeletal sea lions, but a sea lion pup is so much easier to handle than a male skeletal sea lion. But we can appreciate that. And so we were able to crank up our sample size tremendously as well. So we got the cohort of concern, and we can increase our sample number at the same time. And so that was really important. So the Castellini et al. Paper covered mercury in this area. And then Larry Rhea expanded the research into the more concerning areas where skeletal sea lions are not recovering. And so she did this paper in 2013. And this got the attention of NIMS. And we're getting more support to follow up on this. But here's the reason why. We keep adding data to this figure. As you go from east to west, you can see that mercury concentrations climb, and then in this Agatur region, they jump up. And this was a bit concerning, and then they come back down again. So now we have Russian colleagues providing samples, and we'll see if we can work out what's happening. Well, so what? They're bigger. The concentrations are greater. Well, actually, comparing blood and hair showed that hair is a very good matrix for assessing what might be going on in circulation. So they correlate nicely. But also, you can use hair or blood to determine what the risk might be, or put this in some kind of risk context. And so here are guidelines for benchmarks for hair and blood that clearly show that there could be a neurological effect to these concentrations. Some are based on wildlife and some are based on human. But the idea is to put it in the context to say these are relevant concentrations. They're not just higher, but they are moving into the relevant range. And it's a bit of concern. So they're probably coming through the diet. A lot of people want to talk about volcanoes, and a lot of people want to talk about Asia. We don't know. We don't know where the mercury is coming from. We just know the pathway is going to be in the diet. So now the commercial fishing industry and the Department of Environmental Conservation in Alaska are working with us. The commercial fishing industry is providing us fish for free. That's a big deal. And from that region at Western Aleutians. And so now we're looking at what might be going on with the fish. So that's why they're important as sentinels, because management concerns, public health, understanding what's going on in Western Aleutians. I assume John Harley and Stephanie Hughes are known to this audience?
Stephanie.
John Harley? No? Oh, poor John. Oh, John. No, I'm just kidding. John was an undergrad at Santa Cruz that worked with Costa as an undergrad, and then he came up to Alaska because we either brainwashed him or he already was brainwashed, but he was really into the One Health idea. And so we said, well, we can help you. So John's, and this is where our marketing graduate students here, you're interested in this. John gets to work with sled dogs, wolves, and foxes. And so using canids as a model for environmental contaminant exposure, he's looking at gene expression in these animals related to their mercury exposures. And so he's developed chapter 1, he's writing right now for that. Chapter 2 is maybe more relevant to this discussion. Stelis sea lions. He's going to be looking at gene expression in some of the Stelis sea lions from that western Aleutian population, working with colleagues at the University of Washington. And we know that there's some selenium-dependent enzymes that are real important. And John's going to be looking at how those enzymes might be responding, the genes for those enzymes are responding to the presence of this mercury. And that's really sophisticated work. And so he had to work out field protocols for getting the samples, so he had excellent mRNA, and he did. And so not the filter paper, that's what he wants to do next. But using certain technologies, he was able to get mRNA from the field. Then, the final leap, fish consumers in Baja California. We're putting papers into the press right now where people down in the La Paz area have about 5 times higher mercury concentrations in their hair than people in Alaska. They're eating sharks, they're eating other high trophic fish, old fish. And so we've been working with our Mexican colleagues on this, and he's going to take a closer look at what might be driving that mercury accumulation. And that's a big step for John. These two, no problem. This one is one of those risky ones, so he'll fall back on feeding ecology if we can't do the genetic work. But the colleagues in Mexico would like this. Stephanie, who seemed like got more of an applause or anything, she's going to be mostly doing immunology work. And so haptoglobin is an inflammatory indicator, and she's doing this right now where she's looking at haptoglobin in Stelis sea lions and looking at what might drive variation in haptoglobin levels. She's also going to establish baseline immune parameters in free-ranging sterile sea lions that will be used for determining factors that lead to immunomodulation. So what she's doing, like we all have to do in wildlife work, she's establishing the normals before she looks at adverse effects. And so she's got to say, well, what's normal? And I got to consider the biology of the animals. So she's got to look at sex, age, reproductive status, and how that might affect the metrics she wants to use. It's great physiology, biology work. But then we'll come into here and looking at response to contaminants. This is her graph title because we're still trying to get her to sort this out a little bit. But then she's going to look at how contaminants might be involved in modulating the immune system of sea lions. So you can see between Steph's work and John's work, we're trying to get more into adverse effects. We're also doing this with the Marine Mammal Center where we're trying to look at neurodevelopment and harbor seal pups related to their mercury concentrations. So the lab is trying to move with Moss Landing's help, Marine Mammal Center's help, and what we're doing at UF to try and figure out what's going on with mercury in these pinnipeds. I still don't know what she means by this. You're welcome to read it. Maybe Jim can translate. Is that Stephanie's or what?
It's Stephanie.
Decoding cumulative effects of nutrition status, infectious disease, contaminant burden that threaten health status of marine mammals using an in vivo immune functional model. And this one killed me. I don't know, like epigenetics were awesome. That's because she shares an office with John Harley. So I think having those two cook up these ideas is great. And they're both Californians. Thank you very much. We really appreciate it. Got to acknowledge the permits and the boats and the vessels and all the money Noah puts into this. It's a lot of investment to do this stuff. So in conclusion, One Health in Alaska, we have a natural laboratory, if this intrigues you. One Health can advance through simplification of some methods, but must be validated through modern techniques. Alaska and California. I think there's synergism there. I think Jim might agree. And then the whole sentinel species concept, it's true here in San Francisco Bay as well as in Alaska. I think the marine mammals that are in the San Francisco Bay region are important sentinels for a mercury-contaminated environment. I've been making that speech for about 4 days straight now, haven't I? Jim's getting tired. But I'm going to push hard on this because of the concentrations we've seen in the harbor seals and the potential issues that might be popping up at the Marine Mammal Center. I'm inspired to become more aggressive, but I need help from Moss Landing and the Marine Mammal Center make sure I'm not going off and hitting the road. But I think it's time to see where we can go with that. So, thank you very much for your attention.
Any questions? All right, thank you. No, gotta be a question. Ed's got a question. Jillian.
Just a science question, but on that, the very early slide for those polar bears and bears.
Oh, I knew the polar bear. They always
How did they get that picture?
Which one?
How close were they standing to those bears? They're very, I think it's like
They
first or
voted
second.
I can't operate that computer. You mean when they were scavenging the graveyard?
Yeah.
The person, how
They don't care about us.
Oh, wow. They got a lot of bear money, so.
No, there's Zoom on this.
Yeah.
We had to go out there, not for fun and fame, but to assure they weren't getting into trouble and keep people away from them. And then on occasion, not on occasion, quite often we had to haze them. They would get too close to something that potentially result in human conflict. And so we had to haze them back. And we were trying to teach them to land in the sand, or actually gravel, land on the gravel. So that was a part of the interactions. Also, people weren't believing this. Bears don't do that. I'm standing right here. And so we also do a lot of documentation. That's why we went in the helicopter, because we thought there were a lot of bears in the area, but it's like count fish in an aquarium, just drive around there. So we had to go up in the helicopter and survey it real quick. And that's when we counted 60 bears. Unbelievable. Suddenly, Fish and Wildlife Service and USGS was up on the next plane, because they saw a looming problem. Because as soon as there's an unfortunate human incident, it's usually is bad for the predator, right? So they wanted to get up there and do this quickly. So they came up with a 3 strikes and you're out policy, and that was the bear returned to the community 3 times, and then the 3rd time we had to euthanize him. In other words, it was a problem there. It made for a very stressful moment.
Todd, you work in an environment where mercury is accumulating from all over the planet.
That's right.
And through distillation processes, and you say, it's easy to blame the Asians or the different people for. Have you had any luck with mercury isotopes and trying to define where the source is actually coming from for this work?
That we've been working on it for 18 months, and we're trying to write a proposal that would look at mercury isotopes, but the caveats are too numerous, as you know, for how isotopes fractionate the mercury, and so between the sunlight and the feeding ecology and trophic levels, we don't think we can use our fish and marine mammals to do that source determination. We think we've got to go back to the abiotic matrices to do that. Nobody's sampling in that area for that. So it's a real challenge. Okay, what we're talking about is there's many mercury isotopes, and in some ways you can kind of fingerprint the source. However, once sunlight and biological processes have acted on the mercury, that alters the isotopes in a way that you really lose the fingerprint from the source. And so we've been trying to figure out how we could use the samples from the stellars and from their prey, but every time we sit down and work it out, we can't make a strong enough proposal because we end up spending half the proposal outlining the caveats. It doesn't make for a strong argument, right? So if you have any great insight, we'd appreciate it, because I've worked with Canadians, worked with Bloom, and we can't find a way to do it in that level of biota. We have to go back to the primary producers or actual abiotic.
Right, it's like snow or glacial deposits.
Yeah, or sediments. And as you know, we have a big problem in the Aleutians because so much volcanic activity. And so could we actually pick up an Asian mercury signature in that background? Yeah, so it's a very good question, and we are trying to figure out a way to do it.
I noticed from your graph that the mercury levels in the blood were really quite high. I've been looking into the mercury in sharks, especially in muscle tissue. How does the mercury concentration in, say, the blubber or the muscle tissue compare? Is it still quite that high?
Are you talking about the stellars?
Yeah, or I guess in marine mammals in general.
We just put out a paper, and I didn't get it in the presentation, where we actually did, well, we bragging about this president, it's not my job. Tiffany Perkins did a body burden study where she took harbor seals apart and essentially put them back together from a mercury perspective and developed a model of how mercury distributed in harbor seal pups. We went and did the same thing in stellar sea lions, and the majority of the mercury was in muscle and the pelt, just like what Tiffany found in harbor seals. And what we do with body burdens and so on, there you're calculating the concentration in the tissue, but you're also determining the amount of mercury in the tissue. In other words, the actual mass, not the concentration. That's the value of doing this kind of studies. We only did it on 6 stellar sea lions, but it got published because they're very hard to get. And when she looked at the hair of those animals, the hair correlated very nicely with the other tissues. So as hair concentrations of mercury went up, there was a parallel increase in all the other tissues. She looked at muscle, and muscle was a good indicator of mercury concentrations in the other tissues as well. And what Tiffany's paper was really important is we were trying to emphasize that actually liver, kidney, and other tissues are actually very poor at showing the status of mercury because it accumulates in it over their age. So Tiffany showed that muscle is actually a very good way of looking at the overall contamination of that animal. And we repeated it in the stellar sea lions and we got the same thing. So that answer your question? So most of it's in the muscle and the pelt. And the hair and the muscle are probably really good tissues to get an idea of the status of that animal if we can't afford to do the whole 13 or 14 compartments for each animal.
So then the blood seems like it would be a very convenient way to do that. Do those reflect the same levels in the tissue?
We're cautious about blood only because a recent meal high in mercury could cause blood concentration to shoot up and misrepresent what's in the body. That's the only caveat I put there. Yeah, it's more of a transient medium, but if they're really on their diet that they typically are exposed to, then it is useful, and that's what we showed with the stellar sea lions around agitude, was that the hair did parallel what was in the blood in that case. Blood is a bit transient, but does it work most of the time? Yes. And whole blood are working. And we're also getting into studies where we're looking at how selenium and mercury compartmentalize in the blood. That's also becoming interesting because mercury and selenium compartmentalize differently. And if selenium is helping protect from mercury toxicosis, they kind of have to be in the same location. And so we're doing, we did dogs fed fish, we did the bottlenose dolphins, and now we're going to do the stellar sea lions because we want to see how taxonomically these animals compare as to how they distribute mercury and selenium. And the physiologists are really excited about this. So that's another aspect of your question, is that even within the blood we may have compartmentalization. The packed cells, the serum plasma, things like that.
All right.
Send more students.